as researchers, one of the most challenging things to do at the end of the research process is to communicate your findings to an audience. And one of the hardest parts about communicating your findings comes with sharing the data with an audience. I'm going to go through 2 examples of how you can present data to an audience, and I think you'll find one more engaging than the other. After I go through each example, I just want to quickly share a method that you're going to see me apply in one of these examples called QERIP. And it's just a way to present so that your audience can think through the research question with you and sort of build up curiosity for what your data is going to show. So let me go through these 2 examples, and then I'll just introduce you briefly to QERIP. So in these figures, I'm showing that SMAD1, which is a protein, decreases with the treatment of SI-SMAD1. Following that experiment, we were able to measure axons, and what we found is that axons from animals that were treated with SI-SMAD1 showed a decrease in axon length after an injury. In this final figure, you can see an image of the same data that's shown here quantitatively. What you can see is that in the control nerve, axons are much longer than they are in the SI-SMAD1 treated animal's nerve. So this shows that SMAD1 is truly a very important key player in axon regeneration. Okay, this is going to be the second example. The question I was asking in my study was whether SMAD1, a transcription factor, plays any sort of role in axon regeneration. So the approach we used to answer this question was to use small interfering SMAD1, which decreases the levels of SMAD1 protein if the approach is working, if the tool is working appropriately. This figure shows that when mice are given SI-SMAD1 treatment, that compared to control, the levels of the SMAD1 protein are actually decreased. This doesn't give us any answer to our question about whether SMAD1 plays a role in axon regeneration. However, now we know our tool is working. So we can take these animals and induce a crush injury in the sciatic nerve and then watch the axons regenerate and quantitate and measure the length of regeneration in the SMAD1 knockouts compared to the control. So what we can see is if we do the crush injury on the axons, which is shown by the red arrow, you can see that the green fluorescent protein labeled, GFP labeled axons, are those that are regenerating. You can measure the length and count the numbers. So what we saw, you can see in the picture, is that the control axons are much longer than those axons that have the SMAD1 decreased protein. This indicates that SMAD1 must play a role in axon regeneration because the axons are not growing as well after the injury. We also quantitated this, looking at both the number and the length, and that data supported what we saw in the actual nerve when we looked at GFP fluorescence using confocal microscopy. So we are very surprised actually to see that our hypothesis is supported. There are few regeneration-associated genes that have been discovered over the years, and SMAD1 is now able to be added to that list of regeneration-associated genes. There's quite a difference in the way that those 2 figures are presented, and there are a lot of different ways to be more engaging when you present a figure. But I just want to introduce one way to be more engaging, which is to give it sort of like a story plot, as if you're reading a book. And so if you present a figure in a way where you're saying a researcher's story actually is, there's a question, you do an experiment, and you get an answer. And then you interpret that as any researcher does. So what you do is present the figure so that they can see the whole research process in a nutshell. And that allows them to be engaged in the thought process of your experiment and get excited and curious about what the results will be. And so using the Q question, you say the experiment is E, R, you actually give the results, and then I is you're interpreting it, you're telling them what it means. And then P, the problem, is For example, the figure I just showed, I could have explained that because there are axons still growing, SMAD1 is not the only protein involved. There are probably other proteins that allow those other axons to regenerate. And that portion of the QERIP is optional, but it is a very good place at the end of that, using that method and applying QERIP, is to say the limitations that may be there. Okay, so I hope that you'll have an opportunity to use QERIP to make your figures more engaging and your talk more engaging, so that when the audience leaves, they're able to take a strong message with them and not be encumbered by jargon, and to sort of just get excited about the fun and the adventures that you can have in research.